Hi, I'm Jonah Comstock, Editor-in-Chief at pharmaphorum. I'm here at JP Morgan 2023. It's day 3, the rainiest day yet. I'm here at the Donatello Hotel with Prothena CEO Gene Kinney, President and Chief Executive Officer. Thanks for joining me today, Gene.
Thank you for having me. It's delightful to be here, despite the rain.
So tell me a little bit about Prothena. What do you guys do, and what are you here for at JP?
Yeah, so Prothena is a company that spun out from Elan Pharmaceuticals back in 2012. Our focus is really on dysregulated proteins. So as you think about proteins and their need to take on a normal shape in space, a conformation in space, in order to have their normal function, we know that sometimes that process goes awry. So they take on an abnormal shape, they misfold, they take on a misfolded conformation. When that happens, we typically have systems inside cells that recognize that misfolding event and traffic those proteins to the trash system, if you will, inside the cell. But that process can go wrong. And when that process goes wrong, those misfolded proteins have a propensity to aggregate on themselves, to deposit in tissue, and ultimately lead to disease. So what we study at Prothena is how that process actually happens and how we can best intervene in that process. That takes us into 2 areas broadly in therapeutic. The first would be outside the brain. So these are diseases of the periphery. Most notably, when you have deposition of protein in the heart and leading to restrictive cardiomyopathy, you can have a very serious condition up to and including mortality for patients. And so these would be the peripheral amyloid diseases in our portfolio, including AL amyloidosis and ATTR amyloidosis. In the brain, the deposition occurs either in the parenchyma of the brain or inside nerve cells themselves. And when that happens, it can lead to dysfunction of neuronal processes, leading to neuronal degeneration. Here we talk about diseases like Parkinson's disease, Alzheimer's disease. And so we have a number of those therapeutics in our clinical portfolio. Today, we have 5 clinical programs. We expect an IND this year, which will bring it to 6. About half of those programs are partnered with, we enjoy partnerships with Roche, Bristol Myers Squibb, and Novo Nordisk. And about half of those programs are independently owned and are moving forward under our supervision.
So I have heard a fair amount about amyloids in the brain at this show. Actually, I haven't heard a lot about them outside the brain, but let's put that on the shelf for a minute and talk about Alzheimer's and Parkinson's, because I know that is top of mind for a lot of people.
Absolutely.
At this show, there have been some successes, there have been some mixed successes. Where do you think the science is right now in terms of this as a key driving mechanism to for these conditions? Are we pretty sure we're barking up the right tree, and are we doing it the right way?
Yeah, we think there's a couple of proteins to talk about in the context of Alzheimer's disease. Let's start with amyloid beta, because I'm sure that's what you've heard most about. The other protein we can come to is a protein called tau. They're probably on biological pathways, so there's good evidence that amyloid beta will actually cause a dysregulation of tau. So it's not that they're separate ideas. These are actually a similar idea in terms of the biological pathway. I think the understanding of the role of amyloid beta is quite good from a genetic perspective, causal, and we think about mutations in different gene sets that lead to Alzheimer's disease in a causal way. So not associational gene changes, but actually causal gene changes. They tend to be in the pathways that lead to increased amyloid beta production, or in some cases to mutations that lead to a more aggregation-prone form of amyloid. And so we know that amyloid is involved, at least from a genetic perspective, in the context of Alzheimer's disease. What we've seen more recently, and it's very exciting. I mean, if you think about where we've been in the treatment of Alzheimer's disease, it's really been a reliance on symptomatic agents. So treating symptoms of the disease, but not really having an impact on the underlying pathophysiology, the underlying progression of disease. What we're now starting to see with some level of consistency is in a class of, in this case, monoclonal antibodies that target this protein amyloid beta, we're starting to see some consistency that if we can lower in the brain to a sufficient level amyloid plaques, these sticky tangles of amyloid beta that deposit in the brain, we can see a good relationship with positive clinical outcomes. And most recently, our colleagues at ASI have shown with their molecule, a molecule called lecanemab, that depending on which endpoint you evaluate, of course, different endpoints give you different answers, you can see anywhere from about a 27 to 37% slowing of disease progression over an 18-month period as they treat patients with that. They give it a 10 milligram per kilogram dose of that drug every other week, given intravenously. And so we think that that's very interesting. In fact, just last Friday, we saw an accelerated approval by the FDA on that drug. And so I think, we're now seeing that these types of treatments are moving forward as potentially the first class of disease-modifying agents in Alzheimer's disease. So for those that have been studying Alzheimer's for some period of time, it's a very exciting period as we think about patients and the treatment armamentarium that you can bring to patients in the context of what really is a cruel disease. I mean, you think about Alzheimer's disease as a disease that robs you of your memories, robs you of your independence, ultimately robs you of your life, and the impact it has not just on the patient but on the loved ones and caregivers. And I think starting to think about a shift in how we treat patients and moving to the underlying disease pathology is very exciting.
Yeah, on a personal note, I lost my grandmother to Alzheimer's and my grandfather to Parkinson's. So I have a lot of firsthand experience with that patient experience. It is pretty awful.
Yeah, I'm sorry to hear that. Yeah.
So how is what you're working on different or the same as lecanemab that we saw from SI and some of these other ones that they're getting a lot of attention right now because of the approval?
Yeah, so in our portfolio, as we think about Alzheimer's, of course, we have a fulsome portfolio. We have, I'll talk about a vaccine we expect an IND on this year. When you include that into the mix, we have about 6 clinical programs. Within that, 3 of those are squarely focused on Alzheimer's disease. The Alzheimer's portfolio really consists of a couple of different approaches for very specific reasons. As so again, we spun out from Elan Pharmaceuticals. What may or may not be obvious to your viewers is that Elan really was at the forefront of a lot of the science that led to this idea of targeting amyloid beta, particularly with monoclonal antibodies and vaccines in the context of Alzheimer's disease. So having that historical perspective and knowledge base, one of the things that we observed over the years was that the clinical trial design and aspects of clinical trial design and execution were continuing to evolve and improve over a period of time. And as we saw that improvement, the use of biomarkers, patient selection processes to make sure that patients coming into these clinical studies were more likely to respond to these types of treatments than not. And also just thinking about the endpoints that are more sensitive to measuring change over time. We felt like there was a reasonable probability that one of these anti-amyloid agents would actually get across the finish line and become a treatment for patients. As we saw that, we felt that one of the key things that needed to be solved from a patient perspective would be around patient access. These first generation therapies are given intravenously. And while a huge step change in how we think about treating patients with Alzheimer's disease, we still want to make sure that any patient that can be advantaged by these types of treatments has access. So what we endeavored to create, and it's a molecule in our portfolio called PRX012 or PRX12, it is a molecule with a target product profile, which needs to be empirically proven in the clinical trials, but a target product profile of once monthly subcutaneous administration with a convenient dosing paradigm. So, think of a single syringe kind of presentation. The way that you do that scientifically is to create a molecule that can occupy as much amyloid beta as these first generation antibodies, but to do so with a much lower dosing requirement. So we achieved that from a preclinical perspective. That drug is now in phase 1 clinical trials, and we expect to begin reporting data for that program this year. So we're excited about that. That's only one though, and the other 2 actually really come to where is this field going, right? I mean, what we do obviously in biotechnology every day is we think about not just where we are today, but where things are going to go and how do we continue to push that efficacy profile forward. And we and others, so not uniquely us, but we and others in the field believe that with the foundational treatments targeting amyloid beta, one of the things that's likely to happen in the field is the exploration of combination therapy. And that's not unique to neurology. If you think the HIV field, if you look at oncology, the idea of combining treatments in order to push the efficacy profile forward is pretty well established in medicine. And so here we think tau is a very reasonable target. As I mentioned before, these are on pathway. And so we have a molecule in our portfolio. PRX012 is actually a wholly owned molecule. It's completely Prothena owned. Here with this molecule, it targets the tau protein. We're working with our friends at Bristol Myers Squibb. It's a molecule called PRX005, and PRX005 is also in phase 1 clinical studies. So we expect to begin reporting data on that this year as well. And then finally, the other way that you think about this, in addition to trying to push that efficacy profile forward in patients that already have disease, is to start moving the treatment paradigm even earlier. So starting to think about preventative types of studies, secondary prevention, ultimately primary prevention. And there are a number of scientists in the field that are doing wonderful work identifying individuals that have the biological predisposition to develop this disease, but maybe not quite yet the clinical manifestations of the disease. And so in that population, we believe a vaccine may be the right answer. And so what we've actually done there is combine this element of combination approaches in a vaccine. So we have a vaccine called PRX123. And the idea is that this vaccine is designed to generate a titer or antibody response in the recipient to both amyloid beta and to tau simultaneously. So we've actually shown preclinical work on that at scientific meetings. We expect an IND with that molecule this year.
That's super interesting. Now, what I hear is that in neuro, the trick is finding that population, right? One, just enough of them to do a clinical trial, but two, for this to be effective, you've got to, and these are diseases that, like, I don't know, dormant is the right word, but they're genetic diseases that people find out that they have them and start to experience symptoms pretty late in life. So tell me about that piece of it.
Yeah, so you're right. I mean, these are not all genetically caused. The genetic cases, for example, of Alzheimer's disease are a relatively low percentage of the total cases, but they teach us quite a bit about the biology. That's the key. Is if we know something can cause a disease, then we understand that those biological systems are important, even in what we consider to be idiopathic cases where we don't quite know the origin of the disease. Your question about clinical trials is a good one. I mean, the question really is, the observation periods in these studies tend to be in the neighborhood of 18 months. So you're talking about 18 months out of a disease that manifests and continues over decades of life. And so you've got a very short period of very small window with which to measure change over time, and then to measure whether or not you see a deviation, a benefit to that change over time. And so you need very sensitive measures. You need to control variance so that you don't need thousands and thousands and thousands of patients in a clinical trial. And one way that you do that is you try to make sure that the patients that you're enrolling, that you understand how they're likely to progress over, in this case, that 18-month period of time. And that, for example, in the case of an anti-amyloid treatment, that they have amyloid and that they're likely to be good candidates for this type of treatment.
Or you need some kind of longitudinal study or patient registry so you can observe these patients over a longer period of time.
Yeah, right? Ideally both, right? Ideally both. But I think in the context of a randomized controlled trial for the purposes
what you need,
what the regulatory authorities may need in order to adjudicate that you've met the burden of convincing level of effectiveness. I think it tends to be more of those controlled trials, and that's at least where the field is today.
So. Yeah, I mean, obviously, we could get into a whole thing about real-world evidence and decentralized trials and how trials need to change.
Yeah.
I want to ask you my one question I'm trying to ask everybody today about, this conference is always about looking ahead. So what would you say are the biggest R&D pharma trends you're looking towards in 2023?
Yeah, well, for us, I mean, obviously, we're very focused, as we've been talking about, on these protein-based diseases, and we think the field, in particular, in Alzheimer's disease, is in a very exciting inflection point, and it's happening right now. We're seeing now the first 2 anti-amyloid treatments receive accelerated approval. First, Biogen's molecule, and now the Eisai molecule. We have more data sets coming. We'll have another accelerated approval decision coming from the FDA around Eli Lilly's molecule in this space. And then we'll see, we've already seen the phase 3 data set scientifically at scientific conferences from the Eisai molecule, but we'll see now the review of that from the FDA and ultimately the reimbursement then decisions coming from CMS and others. And so we're excited to see how this space continues to evolve. And we think there will be a lot of inflection points around that. For us at Prothena, of course, we're going to be busy talking about data this year. So we'll be talking about data on our PRX12 program. both more characterization around the preclinical aspects of the drug. How does it actually compare on various aspects relative to these first generation antibodies, but then ultimately the clinical data, demonstrating or, at least showing the data to attempt to demonstrate that it achieves the target product profile we set out to achieve. And then ultimately, what is it doing to amyloid in the brains of patients and individuals that receive the drug? And then, of course, for us, as we move into next year, we can look forward to top-line results from a phase 3 study we have in a peripheral amyloid disease. I know we set that aside for a minute, but we have that study reading out next year. That's a drug where, again, this is outside the brain, but in a prior study on a post-hoc analysis, we saw almost, not quite, but almost a 60% relative risk benefit on all-cause mortality. So we're excited that drug is moving forward, and we're excited to see those results next year. And then, of course, we have results coming from our partners at Roche on our Parkinson's program next year. And we'll continue to move those Alzheimer's programs forward, not the least of which is now beginning to work on that vaccine program as well.
Let's talk about the peripheral real quick.
Yeah.
Are there a lot of, this is a similar mechanism. Sounds like it makes sense for you to be in that if you're in the brain version. But I haven't heard nearly as much about, work. There aren't 2 approved drugs for peripheral amyloid. So what's going on there? Is it just a smaller patient population, or are people just not looking where they should be?
These are rare disease populations. And one way that I think about, these diseases as I describe them is, you kind of think about what is the offending protein, where is it made, and where does it deposit and cause it, right? So we've been talking about Alzheimer's disease. We've been talking about amyloid beta. That obviously is a protein that's made in the brain. It deposits in the brain. It causes dysfunction there. Same would be true with tau. A little different place in the brain, but nonetheless. In the periphery, we can also talk about these different proteins. We have a phase 3 program in a disease called AL amyloidosis. The AL really stands for amyloid light chain. The light chain is a protein that's a normal protein. It's made in the body. It's made by a differentiated lymphocyte called plasma cells. And when it's made, it would typically combine with heavy chain in order to make immunoglobulin. So a very important part of your immune system. In the context of this disease, just like, as you might see in multiple myeloma, these are different diseases, but as you might see there, you can get this clone of plasma cells. And unlike multiple myeloma, what's important here is what those plasma cells are doing on the protein. They're overproducing light chain. That light chain doesn't combine with heavy chain appropriately. It binds to itself and it deposits in various organs throughout the body. It can deposit in the GI system, in kidneys, but what really is problematic for patients is cardiac involvement. And what we see is that in the more advanced patients, this can lead to a progressive restrictive cardiomyopathy that can ultimately lead to mortality for patients. And so we've run a prior study there where it was an all-comer study. And really, fortunately, we had stratified based on severity. And what we found was in the more severe subgroup, we actually saw a high level of mortality in our control arm. Median survival was just over 8 months. And in that group, we saw a hazard ratio of about 0.413 at the 9-month time period on all-cause mortality. So with that data, we went to and had discussions with FDA. Ultimately ended up with what's called a Special Protocol Assessment Agreement, or SPA. And there we're moving forward with what's called the AFFIRM-AL trial. It's a confirmatory study to see if we can replicate that effect at an alpha of 0.10. So that's a little bit unusual, but I think it speaks to the interest in this space, and of course that's under agreement with FDA. So that's moving forward. We expect top line there next year. The other peripheral amyloid space.
Is that an accelerated approval pathway?
It is right now. It's a accelerated approval. I'm trying to think. I do believe we have fast track on that. So it would be a potential for accelerated approval. That would be a decision made, I believe, at the time of filing by the FDA. Yeah. The other peripheral amyloid program there is ATTR amyloidosis. Different protein. This protein is called transthyretin. It's made in liver as well as some other places like the choroid plexus and some other places, but primarily in the liver. Its actually normal job is to get the other 4 units. So 4 of them, we call them a homotetramer. So 4 units of transthyretin get together, and they carry proteins throughout the body, like thyroxine or retinol binding protein carries vitamin A around. And when that tetramer falls apart, if those single units aren't cleared efficiently, they actually have a propensity to misfold, bind on themselves, and those can deposit in organs. Now it's going to sound a lot like AL amyloidosis. They can deposit in the heart, for example, or peripheral nerves, leading to disease. There are a number of companies that have approved therapies in this space that actually try to reduce new protein from coming into that pathological process or pathological pathway. What we're doing, which is a little bit differentiated, is attempting to deal with the protein that's already there, the resident protein. So as opposed to reducing new protein coming into the pathway, deal with the resident protein that's already there and causing dysfunction. So that's a program where we have an agreement with Novo, and Novo is running a phase 2 proof of concept study in that space, and so we're excited to see those results when they read out.
So jumping back to Alzheimer's for a moment, Aduhelm and lecanemab have not been without their controversies, and in particular, there have been some side effects, some deaths, and that's gotten a lot of press. There's a lot of questions about how related to the therapy it is and how common it is, but obviously, I'm sure that's something that affects you guys and how you go forward, and at least how you think about messaging and things. So
well, I mean, look, messaging is a result of making sure that you're paying attention to patient safety. Of course, we all care about that. We want to make sure that if there are any real effects or things that one needs to pay attention to related to any given mechanism of action, that we're paying attention to that. We're minimizing the risk of that as much as we can. I think to your point, and I think you're referring specifically to a patient that passed away in an open label extension of a trial, the non-controlled part of the trial. I think the relatedness of that to the therapy is something that still has to be determined. And I think from our perspective, we'll view that from a scientific lens, and we'll try to understand from a biology perspective, whether that's related to this type of treatment or whether it's unrelated or whether it's a factor. And I think, regardless of where that ends up, we'll pay attention to that. We'll make sure that, we incorporate that thinking into our trials. What we can't do, of course, is make conclusions that aren't based on evidence. And so obviously we want to see what the science there continues to look like.
Great. Any other things you've seen at the conference, things that you're excited about? Any final thoughts or thoughts to leave our viewers
with? Well, look, I think, from our perspective, one of the themes that we're seeing is around the CNS and, the neuroscience related therapies. And I think there is a period now where we're starting to see a bit of a renaissance in therapeutics focused on neurodegenerative diseases and, of course, neuropsychiatric disorders as well. I think that's incredibly exciting. I think it's an area where it has untold burden for society at large. You mentioned your family's been affected by it. My family's been affected by it. Living in a time where, quite literally in front of us, we're seeing a shift from only having symptomatic agents in our treatment armamentarium to starting to think about actually impacting the progression of disease and being able to start to combine the thinking around those therapeutic approaches. It's an incredibly exciting time. And, and I'd point to other fields in terms of what that could potentially mean. As we think about HIV as by way of example, AZT being the first therapeutic, there was nobody in the HIV research community who said, well, we're good with AZT, we'll stop there. I mean, it was an amazing step forward in terms of therapy for HIV. But what we saw were better treatments that came, combination studies that came, and now, it's commonly thought that HIV can be a chronically manageable disease. And I think, that as we see those types of examples in medicine, oncology is another good example where we've seen those kinds of approaches take hold. We can start to envision something similar in Alzheimer's. We're right there in the middle of it. So it's a pretty exciting time. And I feel that, and I think, I've certainly felt that at this conference.
Right. Yeah, I mean, I would second that, but definitely neuro is all I'm hearing. And I think, what you said too about this is also happening at a moment when the boomers, the biggest generation, are aging into where they're going to be affected by these diseases. So the timing is good.
Yeah. Well, age-associated diseases are a significant potential societal issue. We have to make sure that we're our healthcare dollars is being spent wisely, and that we don't overwhelm the healthcare system. the Alzheimer's Association has estimated that the current incidence of Alzheimer's disease in the U.S., or I should say prevalence, is around 6.2 million. But that number, without treatments that delay progression, could double by the year 2050, leading to a societal burden of over $1 billion. Alzheimer's Association estimates that in and of itself may be sufficient to bankrupt the current U.S. healthcare system. So, these types of therapies have the potential to subtract burden from the healthcare system. And I think that's how we think about it, is how do we remove burden from the healthcare system, not add to that burden? And these types of therapies from a societal perspective have that opportunity. So that's very exciting for folks like me.
Great. Thank you so much. We've covered so much ground. It's been really interesting. It definitely helped me to understand these protein disorders a little better than I did before.
Great. I appreciate it. It's great to be with you.
Hope you have a great rest of your show.
Yeah, thank you.